In the previous video, we showed that CRISPR experiments can be broken down into 8 different steps. These are guide RNA design, cloning, verification using agarose gels, transformation, selection of transformants, induction of Cas9 and CRISPR gene expression, verification of successful mutant cells, then isolating these cells and checking them using DNA sequencing. In this video, we're going to analyze the costs and trade-offs associated with buying a lot of the equipment to perform these experiments. There are 12 indispensable pieces of equipment required: a microcentrifuge, PCR thermocycler, water bath, UV spectrometer, Bunsen burner, agarose polyacrylamide electropyrolysis gel tank, incubator, UV transilluminator or light box, pipettes, and consumables. The 4 pieces of equipment that matter probably the most and are the most difficult to get hold of are the microcentrifuge, PCR thermocycler, UV spectrometer, and the agarose and acrylamide gel tanks. If you're just looking for a sort of starter kit to conduct your own genetic engineering experiments, then I seriously recommend looking at buying for about $1,800 a sort of DIY biohacking starter kit from a website such as the Odin. This includes a lot of the basic equipment and materials you'll need, such as pipettes, PCR thermocycler, gel tanks. There's the option of getting a centrifuge. They also include a UV transilluminator. The only disadvantage is that the quality of your equipment compared with buying, say, your own centrifuge or your own gel tanks or PCR thermocycler is likely to be lower. But the upfront capital cost is also much reduced, and in terms of the cost of equipment, the value that you can expect is actually quite good. You can even buy a CRISPR starter kit through one of these websites. This includes Cas9 on the guide RNA and an example experiment and plasmid for you to test. There are 2 other potential issues I can see with buying one of these biohacking kits, however. The first is that note that the image that they send on the website is not apparently the same as the appearance of some of the equipment you're getting. Now, I'm guessing this is partly because they always try to source the cheapest equipment possible so they can sort of remain profitable, but this means that the quality of your equipment and the specifications might vary quite a bit. The second big issue, which is in some ways even more important, is getting through a lot of the items in those kits through quarantine if you live in a separate country from the United States, which I currently do. If it's going by plane, airport security officials will not enable you to import that sort of stuff without an extensive permit and being part of an institution, and there would be a lot of legal regulatory hurdles, which I'll talk about in an upcoming video. So now I'm going to just focus on the a bit of a price comparison and some of the crucial features you'll be looking at if you're thinking of purchasing your own microcentrifuge instead of buying it through one of these kits. 2 important things you should consider whenever buying a centrifuge. The first is the maximum rotations per minute, or the maximum rotational centrifugal force. The rotational centrifugal force is a measure of how much force is on its test tube in any instant as it's spinning around in the centrifuge, and the rotations per minute is literally just how many, the rate at which the centrifuge is spinning over time. These 2 measures are essentially separated by the radius of the centrifuge. So larger centrifuges have larger diameters and so require faster spinning rates to achieve the same kind of RCF values. This is a centrifuge from Eppendorf. Eppendorf centrifuges are renowned for being high quality and essentially an industrial standard. The Eppendorf centrifuge has an RCF maximum value of around 14,000 Gs and a revs per minute value of around sort of 16,000, which is sufficient and designed for most laboratory protocols. The drawbacks, of course, to choosing Eppendorf are the cost at around $2,200 and the fact that the centrifuge can only spin 1.5 to 2 mil microcentrifuge tubes. One way of possibly getting around the cost of Eppendorf centrifuges is to buy them secondhand off a site like eBay. As you can see, this is about $1,000 less expensive compared with buying it directly from Eppendorf. Obviously, this comes with the disadvantage of not having a quality guarantee and not knowing exactly what type of condition and how much wear the centrifuge is sustained without inspecting it first. I imagine you might be balking at the cost of some of these large microcentrifuges, so many people buy these mini centrifuges which you can get for under $100. The big problem with these is they don't have the same level of power as the main centrifuges, which means that your laboratory protocols that you're using may not work as intended. Possible compromise you could take is to buy these sort of hybrid centrifuges. These are not as large as the standard centrifuges, but they have almost the same level of power. Eppendorf sells one of these for about $1,500, and you can buy ones much cheaper online from eBay or Alibaba for only around $300 or $400. These should give you similar results to the standard centrifuges, and additionally can be equipped with a PCR test tube holder, which you can use for polymerase chain reaction preparations as well. The next important item which I'd like to talk about are buying PCR thermocyclers. This is Eppendorf's main PCR cycler which they offer to consumers. Now, of note is the fact that they do not directly display the price and you have to inquire, which may mean that you can only buy this as an institution or registered business. Perhaps the most important thing when considering which PCR machine to buy is basically how often are you going to use it and how long do you want your reactions to take. Cheaper PCR thermocyclers and older PCR thermocyclers generally will take longer to go through sort of heating and cooling cycles, which means that your reactions might take a while. However, this is not necessarily a problem if the volume of experiments you're doing is low. As you can see, to buy the centrifuge on eBay secondhand would only cost you about $500, and doing a quick Google search, eBay search of a lot of the centrifuges available reveals sort of a modest price range of between about $500 to $2,000. Important takeaway I think is that if your PCR machine works, your experiments will probably work, it'll just take longer. So if you're doing small scale stuff, buying the cheapest functional PCR machine you can find is probably your best bet. Another item where you might be able to eschew quality for cost effectiveness is buying an Agros electropyrolysis or acrylamide electropyrolysis gel tank. Both of these things are already fairly cheap compared with the other items at under sort of $200 each. One thing to remember is that you also need a power supply. I would recommend not buying any official power supplies from Eppendorf, as these are not functionally that different from the power supply that you would buy sort of for a car charger, and they do almost the same thing. The important thing to check with your gel tanks is whether the electrodes on them are still intact. If the positive and negative terminals still work, then in theory your gel should be able to run just fine. One piece of important equipment where you will not be able to do the same thing is looking at getting a UV-Vis spectrometer. A UV-Vis spectrometer is used to measure the concentration of DNA after crucial steps in any CRISPR experiment like cloning or digestion. Because it has to be calibrated, a second-hand spectrometer may not necessarily measure your concentration of DNA accurately, which means you might have spent a lot of money for essentially nothing. Gold standard for many laboratories is Biochrome's NanoVal spectrometer. This uses only 1.5 to 2 microliters and measures concentrations of DNA on the nanogram scale. The Eppendorf biospectrometer requires much larger volumes for measurement, but can be used for activity testing as well as potentially testing the concentration of DNA. Your spectrometer in general needs to be able to measure things at a wavelength of 160 and 180 nanometers in order to excite any DNA in solution. While Biochrome and Eppendorf don't give you a direct quote, it's pretty safe to assume that you could be paying anywhere from $4,000 to $12,000 for one of these spectrometers. Such an overwhelmingly large capital cost means many of you will likely look to eBay or Alibaba to buy a spectrometer secondhand or new more cheaply. One big problem with this is the spectrometers shown above don't actually excite in the correct wavelength to do experiments on DNA or protein. This is a problem which you're going to have to be careful with, and always contact the manufacturer before purchasing. Based on the cost and the level of risk, I would definitely recommend getting a quality guarantee from whoever you buy something like this from before purchasing. Basically, UV spectrometers are probably the most difficult item to buy for a sort of low-key laboratory because they are relatively expensive, and unlike a lot of the other equipment, you really need them to work with a very high level of quality for them to be worth it. A lot of the other equipment you need can be sourced more easily. Pipettes can be bought quite easily off Eppendorf for a relatively inexpensive cost of around $100, and Alibaba sells them for as little as under $50. If your transform cells express fluorescent proteins and you need a means of visualizing them, many biohacking kits come with UV transilluminators. Alternatively, there are Instructables videos to build a UV transilluminator such as the one that I'm showing here. In order to verify your gene editing experiments, you'll need to sequence your mutations using a DNA sequencing device. You can send your mutated sequences to companies which can sequence the DNA in them at relatively low cost. Alternatively, you can sequence a lot of stuff on your own by buying this mini-ion sequencing chip. This chip costs $1,000 to order from the UK and can be used with relatively few materials compared with other methods to sequence single molecules of DNA. Information from the chip can then directly be loaded via USB port into your computer, showing you the completed DNA sequence. I'm going to end on a final note on some of the materials or consumables you use during your experiments. While most consumables like micropipette tips can be bought relatively inexpensively online, some things like these plasmid mini prep kits from Qiagen and DNA purification kits from Qiagen are proprietary. While you may be tempted to use your own formulation, part of the reason why so many laboratories use these proprietary things despite being so expensive is because they're so reliable. If you go through a lot of materials trying to use your own DIY formulation, you may end up costing yourself more time and effort than if you'd simply bought one of these kits. But it is important to factor in the additional cost of a lot of these materials. So looking at the individual capital costs of all of these materials combined, and probably about half as much of the cost again in just sort of consumables, I would say that to buy a really cheap lab to do all of these experiments, you would have to use the biohacking kits, and even that will cost you about sort of $2,000. And to do this properly and have a lab which is almost sort of academic or sort of industry certified would cost you up to sort of $20,000, and it could be even higher if you want to get some fancier pieces of equipment. Important takeaway from this, however, is that the cost, whether you're an amateur or a business, won't stop you from looking at using this technology and conducting your own experiments. In the next video, we will look at some of the laws and regulations regarding the purchase of a lot of the materials you need to conduct these experiments, as well as laws regarding the handling of GMOs.